Okay, our next speaker is Alexander Platt from Temple University.
Okay, thank you. So I'm going to talk about estimating the ages of singletons and rare alleles. So we start with asking just why are we estimating the ages of alleles? And generally there's two classes of reasons. One is because it's intrinsically interesting. We actually want to know how long an allele has been present in a population. The environment's been changing. Which environments has this actually experienced? If there's been a series of demographic events, range expansions, migration events, we want to know which of these this allele has been around and possibly participating in. But we also want to estimate the ages of alleles because an age distribution of alleles tells us something about how natural selection has been affecting the organism. Right? You imagine a region of the genome under intense purifying selection. Selection keeps weeding out variants before they have a chance to become old variants. We're going to shift the age distribution of alleles in this region to being younger alleles. This is a process analogous to the more familiar allele frequency spectrum, right? Intense selection on a region shifts that distribution to being rarer alleles, and like we use the allele frequency spectrum to estimate things about how selection's acting, we can use the allele age spectrum as well. Why do we care about singletons and rare variants? Well, three reasons. One is most variants are rare variants, right? Just about any population you go to, sampled in just about any way you want to sample it, you go and you sequence it, most of what you're going to find are rare variants, right? Here's an example from the UK 10K. You go sample a whole bunch of British people and you sequence them. What you find is half of your variants are singletons. Right? 80% of your variants you find in fewer than 10 out of 7,000 gene copies, right? It's a massive amount of data. And if you don't have a good way of analyzing them, you've thrown out a massive amount of data. It's not just, though, a big chunk of data. It's a big chunk of data that is enriched for the most interesting variants in your population. It's enriched for phenotypically active variants. It's enriched for variants with significant selection coefficients. These are exciting alleles that we really want to be able to investigate. They also have good statistical properties, right? Rare variants are, by and large, independently segregating variants. This is something that I believe Ivana was touching on earlier. So rare variants have some really beneficial statistical properties for certain kinds of analyses as well. But why do we need a new method, right? We've got essentially two classes of methods existing for estimating the ages of alleles. One is to just look at its frequency, right? A new allele is born and it's rare, and it takes time for it to rise to higher frequency. So an older variant, a higher frequency variant is going to be an older variant than a rare one, right? But if you've got a huge class of your variants that are all singletons, right, there's nothing to distinguish amongst them. Right? And there's very little to distinguish between a singleton and a doubleton and other rare alleles. And even furthermore, if we want to make use of an allele age distribution in estimating selection, but we've derived our allele age distribution from an allele frequency distribution, then you haven't actually added any information to the system. There's another group of methods that work by estimating the time to most recent common ancestor of all the individuals that share the allele. Right? This is really an estimate of the lower bound on the mutation, which must have occurred prior to the ancestor of all these individuals to be inherited by all of them. Right? There's no real explicit upper bound on these methods, but they're also completely inappropriate for addressing singletons. There is no useful measure of the time to most recent common ancestor of one individual. Right? And it's these singletons that is really going to form the core of what I'm talking about here. So what is a singleton? A singleton we can think of as a mutation that arises on the external branch of the genealogy at a particular locus. Right? And it can have arisen any time between now, time 0, the the lower bound on an estimate, and time tc, the time that this branch coalesces back into the rest of the tree and is no longer an external branch. That's an upper bound. And what we're really trying to do here is estimate this time tc. And we're going to do it with a statistic I'm calling maximum shared haplotype. And it starts by observing that at time tc, the ancestors of individuals labeled 1 and 2 here are identical by descent for an entire chromosome surrounding the focal locus. Right? I'm going to blow it up a little. There's your chromosomes. And I would like to stop here and just define IBD for a minute in the way I'm going to be using it. So it's an entire contiguous stretch of something that has shared a common ancestry and not mutated. Right? They're identical and they're descentical. Right, so time goes forward. These two chromosomes are evolving independently and they're accumulating both recombination events and mutation events, both of which are shortening these IBD tracts. And that goes all the way till we reach now, time t0. We take our two chromosomes, we align them, and we find where we overlap in our regions of IBD. This is the maximum shared haplotype. This is the statistic that we're using to estimate TC. This is going to shrink as TC gets older and older. All right, so couple things. In practice, we don't actually know who the closest relative in a population is, so we're going to align our focal individual carrying the mutation with everybody and take the longest haplotype tract that we get out of the population. Also, all of the events that happen on this side of the chromosome are independent of all the mutation or combination events that happen on this side of the chromosome. So looking this way and looking this way, we have haplotype tract lengths that are both independent observations of things we can use to build an estimator of that time to coalescent event. So the rest of the talk's really just going to focus on looking one direction. So mutation at one end, keep going till we find some sort of other event, right? And before we get into how the statistic actually works, I'm going to tell you that it does work, right? Knowing nothing else, we can just simulate a big population, pull out a bunch of singletons from it, ask what this statistic is and what the age of that variant is, and indeed the longest haplotypes are exclusively associated with the youngest alleles. The shorter the haplotype, the older the allele tends to be. So it does work, and even by itself provides power for detecting regions that have singletons, that have age distributions that are different from the neutral distribution. And beyond being able to simply detect these regions, it can quantify parameters about how the age distribution differs from the expected distribution. So it's useful in actually creating estimators too. I'm not going to dwell on this because I'd rather show you something about the sausage inside the statistic. So let's start by the simplest case, sample size 2. The colored dots here represent the event, mutation or recombination, that is most proximal to the singleton that occurred along a particular branch. All right? So we've got a blue one, we've got a purple one. The maximum shared haplotype is the distance to the first one, right? Could be the blue one, could be the purple one. It's going to be the minimum. I'm going to use these cursive letters to denote a distance along a chromosome to the first event on a particular branch. Right, let's keep going. We've got a sample size of 3 here in one particular configuration. It's now got a few more colors. Maximum haplotype sharing will be the shortest distance to either a purple dot or a dark blue dot, or if both of them are shorter than either of those, the longer of the yellow and blue dots, yellow and light blue dots. So we've got a maximum haplotype sharing statistic that is the minimum of these values. You can keep going. You can get bigger and bigger. And the reason I'm showing you all of these is actually to point out 3 unfortunate characteristics of this statistic. So first of all, the distribution thing gets bigger and nastier very quickly as the sample size increases, right? Also, these distributions keep changing every time I change the topology. So every topology has a different property lying underneath it. And it's got a bunch of these max terms in it, the maxima over things. It turns out that minima over these events are pretty easy to deal with mathematically, but the maxima are a headache. So we're going to approach this a little differently. Here's a schematized version of a tree, right? And if we consider just the maximum shared haplotype between our focal individual and somebody in this first A sister clade, we get a max hap sharing that looks like this. It's just the minimum along the A branch, the A prime branch, and the maximum of all the stuff that might come out of this A sister box, right? We can do this for just with the B clade. That's the minimum of what happens along this branch, along this branch, along this branch, and the maximum of what comes out here. And we can keep going until we get to the root of the tree, right? The overall max hap sharing, then, is the maximum over the things that come out of each of these clades. The thing to notice about these distributions of minimal values is that they're almost nesting, and there are more and more terms in them as we go farther and farther down the list. So the expected value of each of these terms is getting smaller and smaller as we're going down the list. And we're going to take the biggest one, which means these ones at the bottom probably aren't really contributing to the value that we're actually going to see at the end. And in fact, it would be nice if we could propose a statistic that is MHS of just the A clade, and we could ignore all of these other terms. And we can test by simulation if this really changes the value we're estimating or not. And it doesn't. There's almost perfect correlation in sort of reasonable populations between the statistic that includes just this term and the one that includes all of these. These guys really don't do anything to the maximum haplotype sharing statistic. Furthermore, this value here is the maximum of several, if it exists at all in a particular tree, is the maximum over several draws of things that are expected all to be larger than A, which means that it's going to be a very unlucky set of draws where this is really contributing meaningfully to this overall minimum value. We can propose another statistic that removes this term as well, and we find that it is just as good a statistic in terms of describing the actual maximum shared haplotype that we're going to find. Could we continue and get rid of this A prime term, which is also slightly hairy? No, that's too far and too greedy. So what we're left with is an approximate statistic that is simply this and only works with this part of the tree, this part of any tree, right? That's the branch, the external branch on which the mutation happens, and the first sister branch before it coalesces, before it gives rise to anybody else, right? This is great for two reasons. One, it's small, it's tractable, we can deal with it. And the other is, right, it means that all of the rest of the tree, which isn't actually involved in this thing you're trying to estimate, isn't affecting your estimator, right? So it's a really comforting thing to see as well, really encouraging. So if we call the length of the A branch T, we call the length of its sister branch phi. We can model this as a Poisson process going along on each branch, mutation and recombination. They're all competing. And the whole distribution turns into a simple exponential distribution with a rate parameter that is the sum of the branch lengths times the sum of the rates, the process rates. That gives us a probability distribution. To turn it into a marginal likelihood, we have to integrate out this phi term because it's not something that you can actually observe in your data. This conditional probability term is not trivial, but it's not something I'm going to talk about either. And you end up with a maximum likelihood estimator, and that really does a very nice job of describing the way this statistic behaves in simulated data. I don't really have time to go into a few extra points, but I'd like to say thank you.
This is great, very exciting. I just had a question about, are you assuming perfect knowledge of haplotype phase?
I am for that description. If you don't know phase, you can make diploids. You can try both phases of your singleton. What you get is a mixture model between this thing and the thing that is a random sample from two unrelated individuals. It should work pretty nicely. I haven't tested it. Fingers crossed.
Okay, great. Thank you very much.